Okay, so welcome to our Integrated Pest Management Academic Seminar Series, which is designed to increase awareness and new research of techniques that advance IPM and its adoption in all types of pest management settings. These seminars are once a month during the academic year, and you can find the upcoming seminars as well as our other events like What's Bugging You First Friday series on our website, which I'll link to in the chat in a minute. So now it's my pleasure to introduce Dr. Ed Straw, an ecotoxicologist who has worked with bees for most of his career. He's extensively studied co-formulants and adjuvants and how they impact bumblebees. He completed his undergraduate education at the University of Bristol, his PhD at Royal Holloway, University of London, and a postdoc at Trinity College Dublin. So it's great to have Ed here, zooming in from across the pond.
All right, thank you, Brian. I guess I'll take it away there. Yeah. So I'm going to be talking today about co-formulants and adjuvants. In American parlance, that would be inert ingredients, and whether or not they are an emerging threat to pollinator health. Specifically, we're going to be talking about bees today, and mostly bumblebees. Um, so today we're going to be talking generally about what co-formulants and adjuvants or inert ingredients are. We're going to move on then to running through an example of bees' exposure to co-formulants and adjuvants, what that looks like practically. We'll talk a little bit about the fungicide Amistar, one of my favorite pesticides, before talking about the diversity of pesticide products available in the UK. We'll move on to talking about how adjuvants and insecticides can have a synergy together. We'll hit the third of the bases with herbicides, talking about Roundup. And then we'll close out talking about a systematic review and a wrap up after 40 minutes of talking. You'll be tired of my delightfully English voice. So let's begin by just getting some definitions out there on the ground. There is a difference across the pond as to how we use language here. So I'm not going to be using the term inert ingredients at all in this talk. Inert is a word that implies that nothing is happening. Hopefully, at the end of this talk, you will agree with me that inert ingredients are not inert. And that we should be using language more along the lines of co-formulants, meaning anything that's in a pesticide other than the main ingredient. So the example here of Amistar, you have the main ingredient being azoxystrobin. That's the fungicide. That's what we'd call the active ingredient. Active ingredients are the thing in a pesticide that does the killing of the thing you're trying to kill. Co-formulants then are just a catch-all term for everything else in the pesticide. It can be things like solvents. So if you mix glyphosate in with water, the water is a co-formulant there. It's acting as a solvent. You can also have things like shelf life increases. You can have stabilizers. Dyes of any kind are co-formulants, but the major classes would be emulsifiers and surfactants. And we're going to talk about surfactants a little bit more in a second. So this is a very visual representation of what a surfactant looks like. On the left, there is a leaf with a tight little droplet of pesticide on it, and that's really, really inefficient. That tight little droplet is actually having a very low surface area, so you're getting very little penetration of the active ingredient into that leaf, and that's just not good. You're not going to get a high level of product efficacy there. The droplet is going to dissolve, it's going to evaporate over time, and you're going to get a really poor level of control. On the right-hand side, that tight little droplet bloop, spread out nicely over the surface of the leaf. That's because there's surfactants or wetting agents in that product. And that really high level of surface area leads to really high levels of penetration into the crop, and you get a really good level of control because of that. That spreading out over things is a little thing that we're going to come back to, a little bit of foreshadowing later for what happens to bees. That's surfactants. Now, this other thing I've been mentioning, adjuvants. Adjuvants in American parlance can at times refer to co-formulants as well, and sometimes people on my side of the pond also describe it as this. In this talk specifically, we're going to delineate adjuvants as just a separate product you add to a tank mixture. So co-formulants are their own thing, a pesticide formulation is its own thing, adjuvants are something that you add in on top of the pesticide product into a tank mixture. So if you're spraying a crop, and you say, actually, for my specific use case, I want a little bit better canopy penetration, you might pour into that tank mixture an adjuvant, a separate product that doesn't contain an active ingredient with the specific idea of changing how that pesticide is sprayed. And that is what an adjuvant is in the way we're discussing it today. So definitions out of the way, let's just run through a little example of how bees could be exposed to coformulants and adjuvants and how that compares against insecticide exposure. So the two different things we're going to be talking about today is one coformulant adjuvant heavy pesticide spray and one insecticide spray. So for the coformulant adjuvant mix, we're using Roundup Pro Active and an adjuvant called Torpedo 2. And then for the insecticide mix, we're using a sulfoxaflor-based insecticide called Closer. And we're just going to compare the two and how bees will interact with them. So when you're spraying this herbicide mixture onto a crop, you could spray something along the region of, this is the maximum level appropriate, of just the coformulants and the adjuvants here. You've got 1.2 kilograms of alkyl polyglycoside, 180 grams per hectare of nitrolial, and 1.6 kilograms of ethoxylated taloamine. So in total, you're talking about 3 kilograms worth of coformulants and adjuvants being sprayed on 1 hectare of land. If we compare that to the active ingredient in the insecticide, you're talking 24 grams per hectare. Now, obviously, an insecticide is incredibly potent, and coformulants by design, inert, are supposed to be low potency, but we're still talking about 2 order of magnitude difference from coformulants relative to insecticides. It's not just how much is in the application, it's also how many times you can make that application. So you can spray a field with adjuvants or with herbicides as many times as you like. There is no label restriction. The only restriction is your sanity and your wallet. The contrast there is insecticides. For this specific insecticide we're talking about, you can only spray it onto each crop twice at most, and each field 3 times at most within a year. You can apply a herbicide adjuvant mixture that's very rich in coformulants at any time of day whatsoever, whereas with the insecticide, you could only spray it when bees aren't foraging. It says, do not apply in the heat of the day during bee foraging activity. So not only is there less of the stuff, not only can you apply it less frequently, you also can't apply it when bees are active. But what about where you apply it? Well, it's actually recommended quite often to spray weeds while they are flowering, because that's when they're most vulnerable to a herbicide. It's just a very energy dependent time of the weed's life cycle. So spraying them with the pesticide then is actually very effective. Obviously, when they're flowering though, bees will be visiting them. Contrast this with the insecticide. You cannot apply insecticide on flowering crops, and you cannot apply it on flowering weeds. And actually, if you're going to apply it near flowering weeds, the label even says spray those flowering weeds with a herbicide. So again, a really, really stark contrast in the amount of pesticide exposure that the bees are going to get. Finally, in terms of plant stage restrictions, you can apply herbicide to any plant stage as long as it's congruent with kind of how you're using the pesticide, whereas you cannot apply insecticides prior to flower development in a way that would lead to residues being in the flower. These are kind of a little bit more relevant in American context than an EU context, because you get glyphosate-resistant crops, and certain crops like soybeans can be sprayed with herbicides during flowering. So let's look a little bit at what the labels say, and actually putting this into a little bit more context. Amistar, one of the ones we're going to be talking about today, that's a fungicide very widely used in the European Union, is actually recommended to spray that onto oilseed rape, that's canola, during flowering. So a fungicide that we're going to talk about today as having potential issues with its coformulants and bees is actually recommended to be sprayed during flowering. That's the most effective time. Same goes for strawberries. Strawberries, incredibly bee attractive crops. They bloody love them. Spray it during flowering. And actually, the information that farmers are given, this is the environmental information sheet that farmers are given for Amistar. It explicitly says for bees, there is no risk management necessary. Amistar is of low risk to honeybees. So we're not blaming farmers for using pesticides wrong or anything like that. The information they are being given is to spray these pesticides at a time that leads to exposure to bees, and they're explicitly being told these substances are safe for bees. So I want to be really clear, we're not blaming farmers here. Let's make this very, very visual though. This is a field in the Netherlands. You can tell because it's flat and it's full of tulips. And it's being sprayed with a boom sprayer, and it's spraying a miticide and a fungicide. As you can imagine, if you've ever been to the Netherlands and seen a field of tulips, this field will be alive with insects. There will be a lot of pollinators in this field, and in that cup-shaped flower, that fungicide is being blasted directly onto whatever is inside that there. So when we talk about the exposure bees have to coformulants and fungicides and adjuvants, we really are talking very high level. There's no attempt whatsoever by regulatory regimes to protect bees from these exposures. Okay, we've laid out why it's important to think about these things because the exposure levels are very high. Let's actually drill into one kind of pesticide and figure out whether or not that one specifically is bad for bees. So we're going to be talking about my favorite, which is Amistar. Amistar came about in the late 1990s. It was the first in its class of QOI inhibitors, very effective, very widespread level of efficacy. It's got 4 main ingredients in the UK and EU. The active would be azoxystrobin, and then it's got 4 coformulants. It is my only conspiracy theory I believe in, which is that pesticide companies deliberately name ingredients difficult to pronounce to make it harder for researchers to work on them, which is why I learned them deliberately. So the 3 coformulants that are listed on the label are alcohol ethoxylates, naphthalene, L-sulfonic acid, and 1,2-benzothiazole 3,2-H1. 2 of those at the end are very, very boring. We're just going to gloss over them every time you see them on screen, but alcohol ethoxylates, that's the important one. We're going to come back to that. So the test we're going to talk about today is working with Bombus terrestris. Bombus terrestris is basically Bombus impatiens, but European. It's got a slightly nicer accent and worse food. And we're working with OECD 247, which is an internationally agreed upon testing protocol. It works for Bombus impatiens, it works with Bombus terrestris, and it's just mortality testing. We're just feeding the bees a single acute dose of the pesticides, so that's oral exposure, and we're just seeing whether or not they die. We jazz it up a little bit with some other metrics, but that's the fundamentals of the protocol. We are feeding the bees a dose of 200 micrograms of active ingredient, or a representative equivalent dose of each of the coformulants based on what percentage they are relative to the active ingredient. We are here just talking about like hazard testing. We're not talking field realism. We're not saying bees are getting this exact dose in reality. We're just kind of talking about this slightly in abstract to kind of understand whether or not coformulants can be toxic. So the methods that we used, we had over 30 bees per treatment. So this is nice and robust. It's more bees per treatment than the guidelines recommend. The 2 controls that we have a negative control and a positive control. So we're really hitting all of the hallmarks of good laboratory practice here. And then we have a series of treatments. We have Amistar, that's just the formulation on its own. And then we have individually each of those listed coformulants and then a mixture of all of the coformulants together. We don't have just the active ingredient on its own here because the active ingredient needs coformulants to be dissolved in water because it's a really difficult substance to work with. That one exciting coformulant, alcohol ethoxylates. These are C16 to C18 alcohol ethoxylates. They make up anywhere between 10 and 20% of the weight of the product, and they do 2 things. They act as surfactants, those are the little bloop chemicals, and they act as emulsifiers too to keep it in a nice, rich, frothy formulation, keep that main ingredient mixed in together. These have never before been tested on bees. I know this because I did a literature search that we'll come to at the end. This is the first time anyone has ever fed bees these specifically and looked at what happened. And the 4 things that we looked at were mortality. After we fed the bees this stuff, did they die? Then we looked at how much food they ate, how much sugar they drank, what happened to their weight, did they gain or lose weight, and then what happened to their guts. So let's start off simple with what the regulatory regime in the EU cares about, which is mortality. When we fed the bees the negative control or 2 of those boring, hard to pronounce coformulants, almost none of the bees died, like one or two of the bees died, statistically non-significant levels of mortality. And our positive control was very successful. We saw 100% mortality in that exposure there. What gets interesting is when you plot onto this graph any treatment that involves alcohol ethoxylates at all. You end up seeing between 20 and 35% mortality for any treatment that includes this coformulant. So it really does look like the active ingredient, the azoxystrobin, is doing nothing, and all of the mortality is being driven just by this one coformulant. Now, regulatory regimes have this kind of thing about 50% mortality. They say that you need to hit 50% mortality at this exact dose that we're using for it to be relevant, for it to pass, for it to fail the testing there. So what we've shown here is that this exposure causes some mortality, but it doesn't hit the threshold for actually further investigation. So 75% odd of those bees are surviving, but actually let's look at the health of those 75% of bees. Like, are they healthy even though they're alive? This is what we would call sublethal metrics. This is a statistically significant effect, by the way, and all of them are going to be statistically significant. So this is how much sugar the bees consumed every 12 hours for 120 hours. That's 5 days. You see an initial spike right at the top there. This is because we starved them for a little bit before feeding them the pesticide to make sure that they would eat it. But then it levels off at about 0.2 grams of sucrose every 12 hours. The bees fed alcohol ethoxylates, or any treatment that involves them, ate about half as much sugar as the bees that weren't fed that. So this is only the living bees. The dead bees were excluded from this data set. So even though they're alive, they're really not hungry. That's, suggesting something is going wrong in their physiology. The fact that they were eating less, that has knock-on consequences for other things. So the negative control on those two boring coformulants, those bees gained weight over the course of the experiment. The y-axis here is change in weight from the start to the end of the experiment, the x-axis being different treatments. Alcohol ethoxylates, on the other hand, those bees on average lost weight, again, statistically significantly. So alcohol ethoxylate, a coformulant, a inert ingredient, is causing bees to die, it's causing bees to lose appetite, and it's causing bees to lose weight. What is driving all of this? Well, it's their guts. This is what a healthy bee's gut looks like. It's yellow, it's a little bit messy. That's a bee's gut. If you decided your gut out, it would look messy as well. This is what a bee that has been exposed to alcohol ethoxylate's gut looks like. I don't know if you can see my pointer, but these big brown sections here are not what healthy guts look like. Thank you, Brian. These are essentially, we are pretty sure they are areas where the gut has died, and then melanin has been deposited there effectively as scar tissue. This is not a bee who died during the experiment, so this is a sublethal effect. The bees who died during the experiment, a lot of them had much worse looking guts than this. But basically what we're seeing here is a coformulant killing off parts of a bee's gut, damaging their kind of appetite and a lot of behaviors that they have, and having negative consequences for them. The gut damage we can quantify by throwing those images into image J and basically mapping them out. Statistically significant effect there. So coformulants can be more important than active ingredients. So the main takeaways from the Amistar section there. Coformulants can be more toxic than active ingredients. I really need to drill that into people. They are not inert. You really, really need to think about them as just another chemical that bees are going to be exposed to, right? A fungicide shouldn't impact a bee. Neither should a coformulant. Think about them both as equivalent in terms of how toxic they could be. Also, regulatory regime will miss sublethal effects if they don't specifically design protocols to look for them. To talk a little bit about some of the work that's happened on Amistar, not done by myself, I was lucky enough to do my PhD on Poshbee, which studied the same pesticides right across Europe in a bunch of different ways, and found that Amistar can have effects in the field as well. So this is bumblebee flower visitation on the y-axis, and then there's the control here and Amistar here. So when you spray a semi-field crop, that's an exclusion cage with a crop of purple tansy, Phacelia tantiflora, in it. When you spray that with water, the bees are more likely to visit the crop than if you spray it with Amistar. So some of those effects I talked about in terms of like damaging appetite or weight loss or mortality look like they're playing out in the field. The same is true in honeybees. You actually see fewer adult workers in honeybee crops that were reared on Phacelia sprayed with Amistar, fewer males as well, and you see the average worker body mass being smaller, matching what we saw in my experiment. The caveat here is that that seems to be dependent on which kind of crop is being sprayed. So buckwheat and floral mixes, you didn't see this effect. So maybe it's mediated a little bit by nutrition. Maybe the effect isn't strong enough to show up in all of the experiments. So there is some evidence, although it's not entirely conclusive, that Amistar can actually lead to real effects in the field. And then we have the lab work backing that up to say actually that is driven by a coformulant. This is done by some wonderful colleagues, Dimitri Wintermantel et al. In Germany. So some of you may know what an LD50 is. An LD50 is the dose at which 50% of the population dies. So if you feed bees Amistar, the dose at which half of those bees die, that's Bombus terrestris, the buff-tailed bumblebee, about 140 micrograms of active ingredient worth of Amistar is needed to kill half of those bees. If you add to that a representative amount of adjuvant, so that's a separate tank product that we're adding in there. If you add in a representative amount of that relative to how much you would put in a tank mixture for spraying something like oilseed rape, that reduces the LD50 by over half. So a lower LD50 means you need less chemical to kill the bees. So this is worse. So not only can the co-formulants in a fungicide be bad, when you actually look at the realistic situation of how that fungicide is going to be sprayed, quite often it's going to be sprayed with an adjuvant, and that makes that even worse. This isn't just true for fungicides. That kind of adjuvant additive level of toxicity is also true for insecticides. So this is work done by Anna Wernick and Richard Odermatt from Germany. This graph is a little bit complicated because it is, but basically they're spraying lambda cyhalothrin in a product called Karate Xeon. And basically the toxicity is referenced against just the insecticide on its own. And then all of these are where they sprayed the insecticide alongside an adjuvant as well. And what you see there is that at times it increases the toxicity by up to double. Again, all statistically significant. So when you're spraying the bees with this insecticide at relatively realistic exposure levels, obviously it's going to kill the bees. It's an insecticide, it'll kill the bees. But when you add in an adjuvant, you get way higher levels of mortality. So when we're thinking about how we regulate pesticides, we can't just be thinking about the insecticides or the fungicides or any pesticide product in isolation. You really need to think about how is a farmer realistically going to use it and what other synergies of stuff are we putting in there that's actually going to change the toxicity. Okay, so we've talked a little bit about how different products can be dangerous. Let's talk about how many products there are. So this is some work I did with Dara Stanley from University College Dublin. And we pulled together all of the data on every different product that's available on sale in the United Kingdom. For context, the UK has 70 million people and a reasonable agriculture sector, but much, much, much less diverse than the American agricultural sector. So the figures I'm presenting to you now, if you just imagine them multiplied by 5 or 6 for the level of diversity that you'd see in an American market relative to what I'm going to show you here. So this is all of the pesticides on sale in the UK. For professional pesticides, that's what you need a license for. There's almost 2,500 of them on sale with 266 different active ingredients among them. Having lots of different active ingredients is good. That's tools in a farmer's toolkit. Having lots of products on sale is a mixture of different co-formulants that makes it really, really, really hard to test and really hard to demonstrate that that is safe for bees and for human health. Amateurs, so that's just anyone can just go to a garden store and buy a pesticide. There's 500 of them on sale in the UK. And adjuvants, there's about 300 on sale of them in the UK. Adjuvants themselves specifically aren't actually toxicity tested on bees at all in the European Union. They are just assumed to not be toxic, and you just let them ride. And there's 300 of them on sale in the UK doing a totally unknown thing to the toxicology of bees. So let's move on to talking about Roundup. I like talking about Roundup. It's fun. It's fun to research. When I publish research on Roundup or glyphosate, I get shouted at depending on what the results are. If I find something that says glyphosate isn't bad, environmentalists shout at me. If I publish something saying Roundup might be bad, farmers shout at me. Let's see what the Q&A section of this looks like. But Roundup's a really good one to work on. It's the trade name for glyphosate. We're going to be talking today about two Roundup products. These are both made by Monsanto or Bayer now. Roundup Ready to Use. I just bought this in a garden center on a little day out. Anyone can buy this. And then Roundup Proactive, that's an agricultural product. It's only available to farmers and horticulturalists. You need a specific license to use it. The methods that we use for this one are fast and dirty. This is an undergrad project. I'm not going to put any level of emphasis on this. This is not a realistic exposure mechanism. What we are talking about is herbicides. Herbicides should not impact bees. Let's just see if in any situation they're going to cause mortality. So it's a spray with the commercial, with the consumer product bottle, just worth of it onto 5 bees in a 5 by 3 inch container to see what happens. It's a herbicide, shouldn't be hurting the bees. This is another Kaplan-Meier chart. So x-axis is time up to 30 minutes, and then a jump to 24 hours, and then survival on the y-axis. Bees sprayed with water, essentially none of them died. Bees sprayed with the professional formulation, about 35% of them died. And bees sprayed with the consumer formulation, about 85% of them died. So something in that herbicide is killing the bees at a relatively high exposure level. Is it glyphosate? Definitely not. Glyphosate impacts a pathway that bees don't even possess. There's no way that it could kill them in 10 minutes. There's certainly no way glyphosate could kill the bees in 24 hours. I knew that that argument wouldn't be enough, so I went out to the garden center and I bought a different herbicide that had the same amount of glyphosate in it, and I sprayed that on the bees, and none of those bees died. So we've isolated out the active ingredient. Glyphosate alone does not kill bees. It's something in the difference between Weedol and Roundup Ready to Use, the consumer Roundup, that is causing those bees to die. We don't know what the difference between those two products is because the labels of those products don't give us enough information to actually say what the surfactants in those blends are. Remember, the surfactants are the things that make things spread out over stuff. Let's have a little look at what the bees look like after they have been exposed. This is a bumblebee that has been sprayed with just plain water. It looks like it's just got out of the shower. After you have like a summer shower in the UK outside, you will see bees looking like this if they get caught out in the rain. Not wildly happy, but it's certainly not going to die from this. It'll groom itself a little bit and it'll fly off and it'll be absolutely fine. This is what a bee that was sprayed with that high level of herbicide looks like. It looks like it's been dunked in hair gel. What we are hypothesizing is happening is those surfactants are causing the surface tension to be so low that the hairs and that liquid is wrapping around the bee's body and maybe penetrating into the spiracles. Those are the breathing holes on the abdomen of the bee, and essentially choking that bee to death. Not a particularly pleasant way to go, but this is with a high level of exposure. We would need to see in the field what it would actually really look like as to in what situation a bee would get this kind of level of exposure. It's relatively hard to do those calculations. But again, I will refer you back to 3 kilograms of surfactant being applied per hectare, being allowed to be applied onto flowering crops during peak bee season. One of the takeaways that we had from this was that the labels that pesticide products have are just not descriptive enough at all. And that's much more of a problem in the US than it is in the EU. So these are the labels of some pesticide products. In the American label, it says glyphosate, and then it says surfactant blend, brackets, proprietary. What the hell am I supposed to do with that information, right? Like, I want to know what in this substance is causing toxicity. I want to know if the ingredients in this substance, in this product, are harmful to humans. And you're just telling me it's proprietary information. What am I supposed to do with that? In the EU, we at least have some regulations that mean that if we found out some of these products are harmful to humans or nature in some arbitrary ways, that they then have to be listed, whereas only other things beyond them can stay secret. So at least the EU formulation lists a couple, but nonetheless we have that catch-all term of and minor formulating ingredients. Minor formulating ingredients doesn't do much for me. Like, I can't find out what is in there without spending, a few £1,000s and breaching EU law. Whereas competitor pesticide companies very, very easily can find out what is in their competitors' products by A, putting it through an LCMS and just not publishing the results, which is perfectly legal to do, or B, hiring someone from their competitor's pesticide team, which they do all the time because they have aggressive, sign-on policies. So this led to me publishing an article called End Co-Formulant Secrecy, which was laying out the argument that we really have no rationale for pesticide companies being allowed to keep secrets as to what is being sprayed on food, what farmers and farm workers are being exposed to when they are spraying these things. There's just no strong rationale for why this should be a secret and explicitly protected under US and EU law. Okay, the takeaways from Roundup. Co-formulants can be more toxic than active ingredients, exact same as the fungicide takeaway. We do need to do a little bit more work on this and see if this actually happens in the field. I would imagine that under field realistic situations, you get much, much less toxicity, but nonetheless, we're still demonstrating that co-formulants can be more toxic under certain situations. And then in that kind of weed all, the red bottle versus the green bottle of consumer Roundup, there's such a massive difference in the toxicity of those two products, despite the fact that those two products are effectively the same thing. If you're in a garden center, you just pick whichever color you like, green or red. There's no real difference in how they're used, but they have wildly different toxicity to bees. So actually, it's really important that we understand what products are on sale and how and why those different products cause toxicity. And given their strong formulation toxicity, it's kind of distressing that there is 262 different formulations of glyphosate on sale, because that's 262 different mixtures of co-formulants going about there. Okay, we're moving into wrapping it up now. So what do we know about co-formulants and adjuvants or inert ingredients? Well, the answer is effectively nothing. In total, as of 2021, there was 19 papers published from 1970 all the way through to 2021. That number's probably closer to 30. There's some really good work coming out of the States since I published this, and there's a couple of papers from the EU on this as well. But really, this is very, very, very little research, right? If you think about every single pesticide application that has ever been made has a co-formulant in it, and a good chunk of them are going to have adjuvants in it as well. And then we have 30 papers, maybe today 30 papers, on how toxic they are to bees, the most important class of pollinators in the world. To put that in numerical context, one insecticide in 2015 had 300 papers testing it. That's 2015, so many years ago, well before the peak of the insecticide research boom, had 300 papers. And that's one single insecticide. And in the UK alone, there are 266 different active ingredients available. So really, we know just about nothing about co-formulants and adjuvants. The breakdown gets a little bit more depressing when you think about it by species. Honeybees, there are 16 papers. Solitary bees, 2 papers. And bumblebees, just the 1 paper, being that Amistar experiment I showed you earlier. Really depressing to think that we have, 20,000 species of bee in the world, and at most 4 of them have ever been tested on coformulants. And the one that we've done the most testing on is the one least in need of saving. So, wrapping it all up now, the exposure that bees have to coformulants is very, very widespread. In the context of insecticides, it is orders of magnitude worse, and that's largely driven by fungicides, but it's also driven by herbicides as well. Coformulants are very, very diverse. We simply don't know what most coformulants are because they are a secret, but the massive diversity of pesticide formulations on sale means that there is a guarantee that there is hundreds, if not thousands, of different coformulants out there that are seeing exposure to bees and to humans in that. Coformulants can drive toxicity. They can be more toxic than the active ingredient. This is not just true for bees. We actually know that a lot of human health impacts from pesticides have been driven by coformulants. The EU has been pretty good at banning a bunch of toxic glyphosate-based herbicide coformulants like POEA or taloamine. So coformulants can drive toxicity in a range of situations. We also know that existing test protocols for pesticides are very poorly suited to measuring pesticide coformulant toxicity. They were designed to measure the toxicity of insecticides, and insecticides drive toxicity in a very different way to things that are not deliberately toxic to bees, but when you put enough of them into the mixture, are toxic. So you bring all of this together, you have a very poor level of research, a very high chance of some level of risk going on there. And you bring all of that together, and it's just like, this is an area that we need to be putting more work into. It's certainly not like the only factor causing bees to be dying. And it's certainly not the main factor, in bee declines. I don't want to over-egg this, but it certainly is an area that's very ripe for further research and something that we could really, really look into a lot better and regulate a lot better. With that, I'd like to acknowledge all of these wonderful people and the funding bodies that gave me money and ask you guys if you have any questions.
Okay, thank you, Ed. That was very very interesting. Yeah, and it made me wonder a few times about differences between the EU and the US in terms of approval. And actually, so there's one question on that, and we can get to that in a minute. But if anyone wants to unmute and ask a question, you can feel free to do so or type your question into the chat. Jeff Scott, go ahead. Go ahead and unmute, Jeff. It's unmuted.
You're unmuted, Jeff.
Might be having some technical issues, but yeah, feel free to type your question in the chat, Jeff. We can't hear you. Yeah, other questions? Otherwise, yep, go ahead.
I do have a question. Thank you very much for the talk. I feel like I'm kind of informed on the topic, but still very surprising to see all those differences in the toxicity. So, Ed, I have a question. Do you know if there's a way to, is there any kind of information that we can have to generalize coformulants toxicity by formulation? So let's say if you have an emulsion compared to a granule or a dust, like are there typical coformulants that are used for different formulations and we can associate the toxicity to them?
You would imagine that some of that contact-based toxicity would not be as likely to be happening with the dusts or the wettable granule formulation type stuff. But beyond that, there's just not enough data to really make an estimate there. You could kind of loosely class coformulants together in terms of like emulsifiers and surfactants, but we do know that different herbicide products have, with both of them having surfactants, still cause very different levels of toxicity. And one of the problems that we have with coformulants is that they're so diverse as chemical groups because you're not really thinking about it like when you develop an active ingredient, let's say an insecticide, you're specifically mostly trying to target the same neural pathway with every different chemical coming together, whereas co-formulants can just a really diverse set of chemistry comes together into them to make them. So I don't think we're even remotely at the level of research where we could have any form of generalization on this. Like we just have such a little amount of understanding, and so much of the research is already out of date as co-formulant chemistry has changed over the years that we're just so far away from any form of generalization, which is a shame, but maybe we'll get there in 20, 30 years.
Thank you.
I have a question for Ed.
Please do, yeah.
Yep, go ahead.
This is a great conference. Thank you so much for working in this field, because I'm a chemist working at Cornell Agritech at Cornell University, and I'm an organic chemist who's been working with surfactants. I've been publishing on surfactants, not in the field of agriculture, and I'm really concerned about the use of surfactants, because as you describe, it's not about only the surfactants that you can find in the product you buy, it's also the surfactants that you can add on the top of that. And what drives me nuts as a chemist is most of the time, all the agrochemical companies, they add adjuvants because none of the compounds are soluble in water. So there is already a ton of adjuvants in the product from the agrochemical. And then on top of that, we add additional surfactants, which, in my opinion, have no use for that. Now, when I try to break down into other chemicals, you just work on the alcohol ethoxylate, but I know that there is like lots of other chemical families like silicone agents, or methylated seed oil. Do you have any other data on other type of chemical family that are found in adjuvants that can be toxic for bees?
Most of the research that has been done on co-formulants has been done on surfactants, and specifically that Wernick paper that I presented did test a few different chemistries of surfactants and found all of them to cause the same level of increase in insecticide toxicity. You can look at it two ways, right? So with the oral toxicity that we saw from the alcohol ethoxylates, that was the co-formulant itself being the toxic thing, and that would differ chemistry to chemistry for different classes of surfactant. You can also look at it as the surfactant doing its job, and then you just care about it being that one class of chemical. So when you think about an insecticide being sprayed with more surfactants in the mixture there, it doesn't necessarily matter what kind of chemistry of surfactant you're using, because all that really matters is the efficacy that you have there. So if that surfactant leads to those droplets clinging to the bee better and penetrating into the bee better, it can be, you know, any different kind of surfactant, but it's still going to be driving that toxicity. Yeah. So there's like 3-4 different classes, different groups of surfactant that I can think of that have been toxicity tested. And there's a 2021 systematic review I wrote that details all of them quite exhaustively.
Thank you.
Great.
Thank you.
Thanks, Ed. So yeah, let's turn to some of the questions in the chat here. First one is about differences between the US and EU. In the US, to get an inert ingredient approved, someone asks, by the EPA, a company must provide invertebrate toxicity data. And so in the US, wouldn't that inform pollinator risk mitigation requirements listed on the product label?
On the product label. So by no means an expert on American regulation. I'm not sure that that's true, though. I really don't think that every co-formulant has to have invertebrate toxicity data to a degree that's relevant, really. If you really, what we're talking about here would be Apis mellifera testing, so that's honeybee testing, and you'd be talking about like acute exposure, oral exposure, following some of the American EPA guidelines there. I have my doubts that all 4,000 identified co-formulants in America, something like that, have gone through that level of testing. And even if they have a lot of that ingredient, if they have, that information is proprietary and held between the pesticide company and the EPA. It's not made publicly available, and it certainly doesn't get published on product labels and things like that. So if you can cite specific legislation, I'd be really, really interested in that, but I'm not sure that that's true.
It's
Okay.
actually on the EPA website for requirements for getting inert ingredients approved.
Okay, but which invertebrates are they spraying on, testing on?
That I don't know.
Because it might be like Daphnia or it might be other things, but the idea that they're doing like for every single inert ingredient having any like relevant level of pollinator testing, I'm not sure on, but I'm really, really happy to look into that. We certainly don't do it in the EU, and the EU has a pretty advanced level. It's like pretty advanced level of pesticide regulation. We do some stuff, but it's just terrible. It's like it's basically worthless.
Yeah, a good example I can give is there are glyphosate products that can be used for terrestrial weed control and some that can be used for aquatic weed control. And the ones used for aquatic can't have some of the surfactants used for terrestrial in the terrestrial products because they are toxic to aquatic invertebrates.
Mm-hmm.
So EPA is looking at the toxicity of inerts before they can be used in a formulation for a given use.
Okay, yeah, that's interesting. Yeah, I can't really speak to the EPA.
And Dan was also asking about the Roundup photo you showed. The label mentioned fast action, and we were surprised because Roundup can take a while to work. Was there another active ingredient in that?
Not in that one, no. That's just like rubbish marketing there.
Okay.
Sometimes they have pelargonic acid in them as well, or acetic acid. You get a really weird, I don't know if you get it in America, there's just been this really weird explosion of Roundup knockoff brands that they're still from Monsanto, but it's like Roundup, no glyphosate, and it's like, well, that's not Roundup, is it? You're just spraying vinegar there, or it's Roundup, it's actually pelargonic acid, and they're just trying to use the brand name again. That specific product we're talking about there, the Roundup ready to use, just had glyphosate in it, and then it had no further information on what co-formulants were in there that were relevant to me, and it certainly didn't have another active ingredient on there because that would legally have to be listed on the label.
Okay. And Jeff Scott lists a paper of interest, Mullen, current opinion in Insect Science from 2015. Not sure if
Yeah,
that's
Chris Mullen is one of the really fantastic researchers on this in the States. Some of his work was really, really seminal in getting me interested in this. Yeah, Chris Mullen, Scott McCart's group, Richard Oderma's group, and Julia Fine, and there's a couple of other people as well that are really fantastic on this. But yeah, Chris Mullen's paper is a real good one to read. I'd recommend it. I cite it in almost everything I write.
And yeah, a general question on what other factors are killing bees. This might be a little bit outside the scope of your research.
Yeah, so I guess when you think about what is killing bees, the most important thing, and I'll always just say this, is that it's just the fact that we're destroying every habitat that they like, right? In the UK, I can't really speak to America, but in the UK, like land is either agriculture or it's a forest or it's, like suburbia. We just don't have wildflower meadows anymore. If you think about American agriculture with these massive, massive fields, there's no hedgerows. Where are you going to put the bees? Really, it's just land use change, I would argue, that is the main cause of bee declines historically. Pesticides certainly play an effect there. There's some good evidence that certain pesticides in the EU knock down bee populations, things like neonicotinoids. There's more broad scale evidence that pesticides generally knock down bee populations or have a suppressing effect on bee populations. But it's mostly land use, with pesticides coming a bit lower than that. Climate change is continuing to have an effect. Invasive species are having an effect, things like Varroa, Asian hornet, all of those things there. But really it's land use. And that's just because of how humans use land for agriculture and for other, purposes like housing. Pesticides are bad, but they're not the worst thing in the world.
Okay, and then a specific question. Do you know what the mechanism of harm is for the organosilicones that have been found to have toxicity to bees? Is it similar to the alcohol ethoxylates?
No, I don't know the answer to that question. No, I'm sorry, I don't know.
Okay. Okay. Is there a link to the page where the EPA describes it? Okay, that Dan can maybe answer this one here and put that in the chat. Link to the EPA. And then Jeff Scott's question from earlier put into the chat. One small correction to suggest the statement that glyphosate cannot kill honeybees because they lack the photosynthesis pathway. This is the target, that is the target in plants, is false. Glyphosate is toxic to fish, for example, and they do no photosynthesis.
So the statement was that glyphosate couldn't kill bees within 30 minutes. There are bacteria in bees' guts. So there's some fantastic work by some researchers in America that found that glyphosate can have impacts on bees, like pure glyphosate can have impact on bees, but it's not going to have an impact in the timescales that we were talking about there, 30 minutes to an hour. Really, really exciting research that finds that glyphosate knocks down specific subgroups of important honeybee microbiome, and that then sensitizes the bees to parasite infections. So while the glyphosate doesn't necessarily cause that high level of mortality, it then leads to relatively common infections becoming actually quite lethal. But yeah, you are correct that despite lacking the pathway, humans lack the pathway of glyphosate, but it can still nonetheless have a degree of impact.